When you were a little kid, it may have seemed like your parents were always telling you to wash your hands. They had a good reason for that, even though they may not have realized it. Your favorite restaurants that warn you about undercooked meats may be helping you avoid more than just food poisoning. A tiny parasite by the name of Toxoplasma gondii may be lurking on your hands or in your food. In this video, we will focus on the parasite's life cycle and its potential link with schizophrenia. Moving on, let's explore a little bit about Toxoplasma gondii so you can see how it spreads and may be causing schizophrenia. The Toxoplasma gondii life cycle is broken up into two phases or stages, a sexual phase in the cat and an asexual phase in any warm-blooded animal like humans. To get technical, this is what happens. In the asexual phase, initially, the parasite invades a cell and forms an intracellular vacuole containing bradyzoites, or the slowly replicating form of the parasite. From here, the slow bradyzoites form tissue cysts in the muscle and in the brain. At this stage, the Toxoplasma gondii is undetectable by the host's immune system since they are hiding in the actual cells. The Toxoplasma gondii reproduces and spreads by endodiogeny, where the parasite replicates inside the parent cell. The sexual phase takes place inside the cat and spreads the parasite all over the place. The cat will ingest the tissue cysts, the bradyzoites, via an infected mouse or the likes, and the cysts will go through the cat's stomach. Eventually, the infested cysts will inhabit the small intestine epithelium inside the cat. Here, the Toxoplasma gondii sexually reproduces and forms oocysts. These oocysts are released in the cat's feces and then ingested by humans. Human infection occurs by not washing your hands after exposure to infected cats, exposure to cat feces when gardening, or by eating infected uncooked meat. So remember, wash those hands. Here's an overview. Eventually, the host cell bursts and a motile form of Toxoplasma gondii, the taxoites, are released. These can be somewhat cleared by the immune system since they aren't hiding inside the innocent host cells, which is good for the host. From here, the asexual phase of the cycle begins again to form the bradyzoites. In the human, the Toxoplasma gondii will enter macrophages in the intestine after ingestion and then travels throughout the body through the blood. The most common sites of infection are the skeletal muscle, heart, brain, and the eyes. For this video, let's focus on the brain. Overall, it seems that T. gondii may selectively affect secretion of cytokines and chemokinases, neurotransmitters, receptors, and/or ion channels. However, further research is needed to confirm this. The effects of infection are almost immediate. After 24 hours, the parasites have replicated at least 4 times. Taxoites produce pronounced inflammatory cytokine response, and so it is hypothesized that the proliferation of these in the brain may be related to the onset of schizophrenia. Serum antibodies are indicative of T. gondii infection. The levels of IgG antibodies significantly increase in infected individuals and serve almost as a T. gondii tag. When a human is infected, they show abnormally high levels of dopamine, as well as increased cytokine production. High levels of dopamine have been correlated to novelty-seeking behaviors in rodents and may explain why they are not afraid of the scent of a cat. In humans, this has been correlated with schizophrenia and bipolar disorder. It has been recently found that T. gondii contains tyrosine hydroxylase, an enzyme necessary for the formation of L-dopa, the precursor for dopamine, and the cyst formations in the brain result in increased synthesis of L-dopa, which may be used for the formation of cyst cell walls or to increase reactive oxygen species in host cells. Cytokine counteracts the dopamine imbalance in humans, resulting in a decrease of dopamine. Since a strong immune response is necessary to fight off T. gondii, immunocompromised individuals, such as those with AIDS, are more likely to exhibit psychotic symptoms. As we learn more about the relationship between T. gondii infection and schizophrenia, better treatment options may be developed. Currently, 3 strains have been isolated. Type 1 has been correlated with acute toxoplasmosis. Type 2 was isolated from mice and is used for rodent studies. Infection with this strain results in toxoplasmic encephalitis. Type 3 was isolated from AIDS patients. By now, you may have asked yourself, if T. Gondii are so abundant, why is schizophrenia a relatively rare disorder? This may be due to two factors. The first is a type of strain which may influence the outcome of clinical illness. Type 1 is a much more aggressive strain in cells and fibroblasts, so it may contribute to schizophrenia. It is believed that type 2 is most likely the cause of schizophrenia since it produces the characteristic physiological and mental changes such as delusions, hallucinations, other psychiatric symptoms, increased retrovirus activation, hydrocephalus, increased ventricular size, and cognitive impairment due to low prefrontal activity. The second factor is response to infection. If there is high cytokine activity or prescribed antipsychotics, the parasite will go into a dormant state. As of now, once T. Gondii have infected, they cannot be removed. As unfortunate and scary as this is to us all, we hope to have gotten at least one message across. Cook your meat and wash those hands.